Okay, so for our exogenous pathway, I'm not actually sure if that's what we're calling it. Yeah, so for the exogenous pathway, this is dealing with exogenous antigens, so we're talking about extracellular pathogens basically. I just wanted to show you that. So let's go back. So these are pathogens, unlike viruses, that actually infect the cell, so therefore they don't need to be endocytosed because they're already inside of the cell. Our extracellular pathogens need to be endocytosed. So there are various different ways that we can take stuff up. Of course, we can do phagocytosis, penocytosis, that's of fluids. Phagocytosis is just endocytosis but carried out by phagocytes, so for example macrophages, dendritic cells, neutrophils, but also it can be carried out by, for example, our B cells. B cells are essentially phagocytes because they can, on the surface of the B cells, they have their BCR, their B cell receptor, which obviously has those fragment antigen binding regions, and so a specific antigen that has affinity for that binding site, that Fab region essentially, can bind to the BCR, and this can be endocytosed, and that's known as receptor-mediated endocytosis. But anyways, however we're taking it in, it needs to be internalized. The pathogen, the extracellular pathogen, needs to be internalized. And what happens is we form something called an endosome, and as it moves further into the interior, it becomes an acidic environment inside of this endosome. And so the pH obviously decreases, as you can see here, and it's become an acidic environment. And so what happens now is that it's going to bind with something called a lysosome, which hasn't been shown here. But if you see the difference, you see that we have loads of dots now, red dots, and before we had just one red stringy thing. And that's basically the antigens from the pathogen that we've endocytosed, but the step between here would be the fusing of the endosome with a lysosome. And this acidic environment of the endosome will activate the proteases and the hydrolases and the other hydrolytic enzymes contained within the lysosome, which will actually break down the proteins and produce the peptides that we need, ultimately to load into the MHC class 2. But there are a few steps before we can load it. So we've produced our peptides, and what we need to do is, at the same time, we need to produce our MHC class 2 molecule. So here we're in the endoplasmic reticulum, and we've got the alpha and beta chains, right? One alpha chain and one beta chain, because remember the structure of MHC class 2 is different from MHC class 1, where MHC class 1 was made from one alpha chain and one beta 2 microglobulin, whereas in MHC class 2 it's made from one alpha chain and one beta chain. So what happens is, once we've produced that MHC class 2 within the at the ribosomes moving into the endoplasmic reticulum, we don't want it to bind to any peptides in the endoplasmic reticulum. That's what these purple things are meant to represent. And so in order to prevent the binding of these peptides in the peptide binding cleft of the MHC class 2 molecule, we have something called the invariant chain. And this is, like I've said, just prevents the peptides from being loaded. Because we don't want them being loaded here. We want them being loaded with the actual, peptides that we've produced here within this separate vesicle. So we then form, this is then going to produce, so this invariant chain bound to the MHC class 2 is going to produce a separate vesicle, and this is known as the MHC class 2 invariant, the MHC class 2 compartment. And within here, what will happen is it's also containing things like cathepsin L and cathepsin P, as well as other proteases. And what they do is they basically cleave the invariant chain. Not completely, not till we get this, there's a step missing here. We're just going to cleave it a little bit so that the invariant chain is still present. And then, of course, the invariant chain is cleaved more, leaving what we can call a clip. and this clip fragment is only covering the peptide binding collector. It's not covering any other part of the MHC class 2 molecule. So again, it's just supposed to be blocking the binding at the binding site of the MHC class 2. Then what happens is it's going to this MHC class 2 compartment basically fuses with the phagolysosome, or the endosome that fused with the lysosome, which contains our peptides now. Those two are going to fuse together, which I suppose would be represented by this, but it's not a very good diagram because it's missing some steps out. But pretend it's a phagolysosome that contains our peptides that were produced in the slide before, and also we have our MHC class 2 molecule, which is currently still being blocked by CLIP in the way. Now, in the within this vesicle, within the cell surface, there is something called HLA-DM, and this is basically going to help to remove the CLIP and therefore allow loading of the actual peptides from the exogenous antigen from our extracellular pathogen. And therefore we've loaded the MHC class 2 molecule with that specific peptide, and it can therefore be exocytosed and shown on the surface and presented on the surface of this antigen presenting cell, and will come into contact with a TCR that's specific for this combination of the MHC class 2 and this particular peptide. And these specific T cells will be CD4 positive. So these are going to be T helper cells.